Welcome to our deep dive, into the world of fragment-based drug discovery. Today we're going to be exploring how scientists are building new drugs piece by piece, almost like assembling a puzzle, starting with these tiny chemical pieces called fragments.
Yeah, it's a really clever approach that's been changing how we think about discovering and developing your medications. If you think about it, most drugs work by interacting with very specific proteins in our bodies, and these proteins are like these tiny machines with very specific shapes. And the drug molecule, we call it a ligand, needs to fit just right in a certain spot on that protein to have the effect that we're looking for.
Okay, I think I'm starting to get the picture. So instead of searching for a fully formed drug molecule right from the beginning, scientists start with these smaller pieces, these fragments, and see how they interact with the protein.
Exactly. It's a bit like starting with, those corner and edge pieces of a puzzle, right? And building from there. Traditional high-throughput screening, or HTS, is kind of like rummaging through a warehouse full of complete puzzles. It can be very overwhelming, and you might miss those crucial pieces. Fragment-based drug discovery is much more targeted.
Wait, so you're saying they don't even start with a complete drug molecule?
Yeah.
That's really fascinating. How do they even know where to begin if they're just working with these tiny fragments?
Well, that's where the real ingenuity comes in. Scientists use a variety of techniques to screen these fragments and see which ones actually bind to that protein that we're targeting. And they don't necessarily need to find a perfect fit right away.
So they're not looking for love at first sight with these fragments.
Not exactly. Think of it more as a first date situation.
Hmm.
You're looking for a spark of interaction that you can then build upon.
I like that analogy.
Yeah, these initial fragments might bind weakly, but that's okay. The beauty of this approach is that scientists can then optimize these fragments, tweaking their structure bit by bit to improve that binding and ultimately improve their effectiveness.
That sounds amazing.
Mm.
But I imagine linking those pieces together isn't always straightforward.
Mm.
What are some of the challenges that scientists face in that process?
You're right. It's not always just a simple snap them together scenario. Sometimes linking those fragments can change their overall shape in unexpected ways, affecting how they then interact with that protein. Other times, the link itself might be unstable or it might interfere with the drug's ability to actually reach its target in the body. So it's a delicate dance of chemistry and structural biology.
So it's kind of like trying to fit puzzle pieces together, but those pieces can warp or break apart if you're not careful.
That's a great analogy. It really highlights the need for careful design and optimization at every single step. And that's where those powerful imaging techniques you mentioned earlier come in.
Right? We were talking about X-ray crystallography and NMR before we started recording. Can we dive into how those techniques are used in fragment-based drug discovery?
Absolutely. X-ray crystallography allows scientists to get a higher resolution three-dimensional view of the protein-ligand complex. It's like taking a snapshot of the puzzle pieces perfectly fitted together.
Okay, so you get this 3D snapshot, but how do scientists use that information to actually guide the design of those better fragments? Are there specific things they look for in those images?
There are. They're looking for clues about how that fragment is interacting with the protein at the atomic level. Where are the hydrogen bonds? Are there hydrophobic interactions? Are there any areas of what we call steric clash where atoms are bumping into each other? All of this information helps them to understand why a fragment binds well or why it doesn't, and how they can actually tweak its structure to improve that interaction.
So it's like having a blueprint for building the perfect puzzle piece.
Precisely. And NMR spectroscopy complements X-ray crystallography because it provides information about the dynamics of the interaction. It's like watching the puzzle pieces kind of wiggle and shift as they interact, revealing how stable that interaction is and how it changes over time.
So X-ray crystallography gives you the static picture,
Yeah.
and NMR shows you the moving.
You got it. And by combining these techniques, scientists can gain a really detailed understanding of how these fragments are interacting with their target protein, which is essential for guiding the design of more potent and effective drugs.
This is all incredibly complex, but also really elegant. I'm starting to see the power of this fragment-based approach. But all this talk about puzzles and blueprints makes me wonder, are there real-world examples of drugs that have been developed using this method?
Oh, absolutely. There are some remarkable success stories that really highlight the power of fragment-based drug discovery. One classic example straight out of an introduction to medicinal chemistry is the development of captopril, which is a medication used to treat high blood pressure.
Okay, tell me more about that. How did fragment-based drug discovery play a role in developing captopril?
Well, researchers started with a simple dipeptide fragment that's just two amino acids linked together. They knew this fragment bound weakly to the target enzyme, but it wasn't potent enough on its own to be a useful drug. So they use a combination of X-ray crystallography, NMR, and other techniques to systematically add more fragments, essentially growing the puzzle piece outward until they arrived at the structure of captopril.
Wow. So they literally built the drug molecule step by step, testing and refining each modification along the way, guided by these imaging techniques. That's incredible.
It really is, and captopril went on to become a groundbreaking medication, and it really demonstrated the potential of fragment-based drug discovery to create effective treatments for really serious diseases.
So are there other examples of drugs where this approach has been successful?
There are ADM-enabling technologies in drug design and development. It describes how understanding drug transporters, which are proteins that help move drugs throughout the body, played a key role in optimizing the antibiotic cefadroxil. They were actually able to use information from studying what we call knockout mice that lack a specific transporter protein to understand how cefadroxil moves through the body.
Wait, so by studying mice that were missing a specific transporter protein, they were able to figure out how to make the drug work better in humans. How does that work?
It's a fascinating process. By comparing how cefadroxil behaved in these knockout mice versus normal mice, they could really pinpoint the role of that transporter protein in the drug's journey. This revealed crucial details about how cefadroxil is reabsorbed in the kidneys and how much of it actually reaches the brain. This is all vital information for ensuring the drug's effectiveness and safety.
That's amazing. So they're not just building the drug molecule itself, but they're also figuring out how to optimize its journey through the body, almost like planning the best route for a delivery truck.
Exactly, and this kind of optimization is essential for making sure the drug reaches its target at the right concentration and with minimal side effects.
I'm really starting to grasp the incredible complexity of drug discovery. It's not just about finding the right puzzle pieces, but it's also about understanding how they fit together, how they move, and how they ultimately reach their destination.
You got it. And that's just the tip of the iceberg.
Oh, I have a feeling we're just getting started. This is already blowing my mind.
Yeah, it really is a multifaceted process. We've talked about, building drugs from fragments, optimizing them, and even understanding how they're distributed in the body. But what about potential problems? Could these fragments or the drugs built from them have, unexpected side effects?
That's a great point. It sounds like there's so much to consider when developing a new drug. Are there any kind of red flags scientists look out for during the process?
Absolutely. One crucial aspect is considering what we call reactive metabolites. As drugs are metabolized by the body, they can sometimes produce byproducts that are, let's say, a bit unpredictable.
So these are like unexpected puzzle pieces that don't quite fit in, and if you force them in, they can actually damage the puzzle.
Exactly. And just like forcing in that extra piece could damage the puzzle, these reactive metabolites, if not carefully considered, can sometimes lead to unwanted side effects or even toxicity.
That makes sense.
Yeah.
So how do scientists identify these reactive metabolites? Are there specific tests or experiments they do? And, what kind of modifications can they make to the drug structure to avoid these problems?
Well, ADME-enabling technologies in drug design and development really highlights the FDA's emphasis on early assessment of these reactive metabolites. It's like checking for potential troublemakers before completing the entire puzzle. Scientists use a variety of in vitro and in vivo tests to see if a drug or its fragments produce these reactive metabolites.
So they're testing early and often to catch these potential issues. What happens if they find a problematic metabolite? Do they have to scrap the whole drug?
Not necessarily. Sometimes they can modify the drug structure to reduce or eliminate the formation of that reactive metabolite. This might involve changing a functional group, tweaking the molecule shape, or even adding a new piece altogether.
So they're constantly problem-solving, going back to the drawing board and fine-tuning those puzzle pieces to make them safer and more effective.
Precisely. It's an iterative process, and sometimes it takes a lot of trial and error to find that perfect balance.
This all makes me think about that time I had to take, antibiotics. I never really considered how complex those little pills actually are.
Yeah, it's easy to take medications for granted, but each one really represents years of research development and careful optimization. And even when a drug makes it through development and is deemed safe and effective, there are other factors that could affect its effectiveness.
Like what?
Well, one fascinating aspect is the concept of polymorphs. These are different crystal structures of the same drug.
Wait, so even if you have all the right pieces, how you put them together can still matter. That's wild.
It is, Evaluation of drug candidates for preclinical development points out that these different crystal structures can significantly impact a drug's properties, like its solubility and absorption. It's like realizing that arranging the puzzle pieces one way makes them fit more snugly and hold together better than another arrangement.
So scientists need to not only find the right fragments, but also make sure they come together in the most stable and effective way possible.
Absolutely. Polymorph screening is a crucial step in drug development. Scientists use various techniques to identify and characterize these different polymorphs, ensuring they select the most suitable one for the final drug formulation.
So even though we've made all these incredible advancements in drug discovery, it sounds like predicting the ultimate success of a drug built from fragments is still incredibly challenging.
You're absolutely right. There are countless factors at play, from subtle changes in molecular interactions to the intricate workings of the human body. It's a complex puzzle with many moving parts.
It really highlights the incredible dedication and expertise of the scientists who are working so hard to develop these life-saving medications.
I couldn't agree more. They're constantly pushing the boundaries of science and innovation to improve human health.
Speaking of innovation, earlier, we touched on artificial intelligence in drug development as a source.
Yeah.
I'm curious to explore this further. Are these new technologies changing how we solve the drug discovery puzzle?
That's a fantastic question, and one that deserves its own deep dive. The use of AI in drug discovery is rapidly evolving, and it has the potential to really revolutionize the field.
I'm all ears. Tell me more about how AI is being used in drug discovery.
Well, AI algorithms can analyze these huge amounts of data, identify hidden patterns, and even predict how a drug might behave in the body, all at a speed and scale that surpasses human capabilities.
It sounds like something straight out of science fiction. Is this technology already being applied, or is it more of a future possibility?
It's happening right now. While the field is still in its early stages, there have already been some really remarkable achievements. AI is being used to screen these virtual libraries of millions of compounds, identify promising drug targets, optimize lead compounds, and even predict potential side effects.
Wow, that's mind-blowing. I never realized how much AI is already impacting the drug discovery process.
It's a game changer for sure. And by combining the power of AI with fragment-based drug discovery, scientists are really poised to make even greater strides in developing new and innovative therapies.
That's incredibly exciting. I'm eager to see what breakthroughs emerge from this fusion of cutting-edge technology and scientific ingenuity.
Me too. It's an exciting time to be involved in drug discovery.
It sounds like we've covered a lot of ground in our exploration of fragment-based drug discovery. We've gone from building drugs like puzzles to understanding the challenges of reactive metabolites and polymorphs. But before we wrap up, I'm curious to hear your final thoughts. So what's the big takeaway, for our listener who's now had this incredible introduction to fragment-based drug discovery?
Well, I think the key takeaway is that fragment-based drug discovery really represents a paradigm shift in how we approach drug development. It's a move away from this brute force screening towards a more rational and targeted approach, guided by a deep understanding of these molecular interactions and, an ever-growing toolkit of these powerful technologies.
That sounds revolutionary. But I'm also hearing that, it's not a guaranteed success. Are there cases where this approach just doesn't work?
Yeah, that's a great question. And you're right, it's not a magic bullet. There are certainly cases where fragment-based drug discovery faces challenges. Sometimes the fragments just don't bind well enough to the target, or they're very difficult to link together. Other times the resulting drug molecule might have, what we call poor pharmacokinetic properties, meaning it doesn't get absorbed or distributed very effectively in the body.
So it's not like a one-size-fits-all solution.
Exactly. It's another tool in the drug discovery toolbox, and its success really depends on a whole host of factors, including the nature of the target, the availability of suitable fragments, and the ingenuity of the scientists involved.
Well, this all underscores the importance of continued research and innovation in this field.
Yeah.
The more we understand about these intricacies of drug-target interactions and the nuances of human biology, the better equipped we'll be to design these safer and more effective medications.
I couldn't agree more. And that brings us back to, the potential of AI and computational methods, which I think are going to be huge game changers in this field.
It's exciting to think about all the possibilities. AI seems to have this huge potential to revolutionize so many aspects of our lives, including how we discover and develop new drugs. Imagine being able to virtually screen millions of potential drug fragments, optimize their structures, and even predict their potential for side effects, all before setting foot in the laboratory.
It's incredible to think about, isn't it? And it's not just science fiction anymore. The progress being made in this area is really remarkable, and I'm very confident that this convergence of fragment-based Drug discovery with AI and computational methods holds a lot of promise for the future of medicine.
Well, on that note of optimism and scientific advancement, we'll have to wrap up our deep dive into the world of fragment-based drug discovery. I think our listener is walking away with a newfound appreciation for the ingenuity, the dedication, and the sheer complexity involved in bringing new medications to the world.
Absolutely. It's such a fascinating field that's constantly evolving. And I encourage our listeners to, continue exploring and learning about it. Who knows, maybe someday they'll even be part of the next big breakthrough in drug discovery.
And on that note, thank you for joining us on the deep dive. We'll see you next time for another fascinating exploration into the world of science and discovery.